This video covers the IGCSE Biology Objective 5.1.7 on the 2025 CAIE syllabus to explain the specificity of enzyme action. So we've already talked about this a little bit, but we're going to explain in more detail. Active sites are complementary. So when we take a look at a substrate and the active site of the enzyme, we see, well, at our level of biology, what we're going to be looking at is the shape. But in essence, as we saw in the previous video, it is actually chemical shape that is going to fit into the chemical shape of the protein, the enzyme, of that active site. But for us right now, we're just going to deal with just shapes like triangles, squares, circles, that kind of thing. So the shape of the substrate must fit perfectly into the active site in order to form the enzyme substrate complex, thereby allowing the chemical reaction to occur, and then the products can form and be released from the enzyme. So we've covered this. But what does this complementary actually do? Why is this important? Why does this create a specificity for that particular enzyme? So not only are they complementary in shape, as we've discussed, but also chemical properties. So they're going to want to adhere to each other. And we saw that in the previous video in the last little slide that we took a look at. So enzymes are specific. So this complementary nature of the active site allows for enzymes to be specific. So if we take a look at these two enzymes and the substrates that are available for them to bind to, we can see, based just solely on the shape, that only certain substrates are actually going to fit to bind perfectly because of their shape into the different types of enzymes. So the pink and the blue circle substrates are only going to fit into the light gray colored enzyme depicted here, versus the orange square and the green, whatever shape that is, fit into the darker gray enzyme because of the specific shape. Those shapes are complementary. So therefore they can only fit into specific enzymes. It's not every enzyme is going to work with every substrate. So due to this complementary nature of the active site, only specific substrates or substrate is capable of binding to a particular enzyme's active site. So in order for an enzyme to break sucrose into glucose and fructose, you must have that specific enzyme that's going to match the shape of the sucrose. And the same goes for any substrate that needs to be split or combined together. You must have a specific enzyme for that specific chemical reaction. This can be looked at and is sometimes discussed as a lock and key analogy. So an enzyme and a substrate is like a lock and a key, where the key is the substrate, and we all know how keys work. Keys fit into the lock, and then the lock will turn. If you have the wrong key in the lock, then the lock will not turn because of the shape of the key and the shape of the lock. So if we think of the key as a substrate and the lock as the enzyme, okay, if the correct key fits into the lock, if the shapes match at all the correct points, then we will have a reaction. Then the lock will turn, or then the enzyme will be able to do the chemical reaction. If we have a key that is similar in shape as depicted in the lower portion of this diagram, but not quite identical to the lock, then we can see here in that final picture that the lock will not be able to turn because all of the points of contact of that key are not fitting in correctly with the lock. And the same is for an enzyme. The key, which is the substrate, which doesn't correctly quite match the lock or the enzyme, a reaction will not occur. Why? Because all the points of contact of the active site are not being bound together between the enzyme and the substrate. So this is an analogy, a comparison that can be used to describe how and why enzymes are specific one enzyme specific to one substrate or a group of substrates in order to produce one specific product.